Dr. Jerry Crabtree here as the speaker for the first Bruce Free Endowed Lecture Series. Dr. Crabtree received his MD at Temple University and then trained in anatomic pathology and pathology at Dartmouth Medical Center. And interestingly, in his part-time while he was a resident, he also developed a research program and generated a variety of high-impact papers, including several in Nature. So that's something that pathology residents like this can aspire to. He next moved to the NCI from 1979 to 85, where he became a tenured investigator. He carried out landmark work on genes involved in the acute phase response and actually cloned the 6th, 7th, and 8th mammalian genes that had been cloned worldwide. And also cloned a variety of other genes, including the human protein C gene, which is also involved in coagulation control and is later developed into an important pharmaceutical in the pharma market. Throughout his career, he has had a long-term interest in immune cell regulation, and while at the NIH, also regulated the gene for what we then called the T-cell growth factor receptor, the IL-2 receptor. At NCI, I joined his lab to learn the new science of molecular biology, which was certainly not KIPS in those days. And it was an exciting time to be in Jerry's lab. And there was also neighboring labs of other future giants, in addition to Jerry, in the field, such as Stan Korsmeyer, Peter Howell, and others. He was recruited to the Department of Pathology at Stanford in '85, rapidly rose through the ranks, including appointment as a Howard Hughes investigator 2 years later, and then a promotion to full professor within 4 years. He was elected to the National Academy of Sciences in '97. And he has received a myriad of awards, including the Warner Lambert Parke Davis Award, the NIH Directors Award, the Outstanding Inventor at Stanford University Award, and the Thomson Scientific Laureate in Chemistry with Stuart Schreiber. So I did a citation search on him, and other people apparently read his papers, since there's over 29,000 citations. And there's a new value called an H index, and he has one of 84, which I'm told is very good, but I don't know what it means. While in his laboratory, Jerry told me that there would be many Nobel Prizes in the field of gene regulation over the coming decades, and of course he's right. And a major general focus of his laboratory has been and continues to be to understand how signaling events control gene expression. In studies on the calcineurin NFAT pathway with Stuart Schreiber, he uncovered the broad outlines of one of the first known pathways that transmit information from the cell membrane all the way to the nucleus. He has made important contributions to our understanding of regulatory mechanisms in immune cells, particularly T cells, lymphocyte calcium signaling, differentiation control in a variety of cell types, including neural cells, including amyloid aggregation events, which are important in Alzheimer's disease, as well as other areas which I don't really have the time to tell you about today. In addition to regulatory events involving transcription factor complex interactions with DNA, he's also studied chromatin events in gene regulation. And so today he will speak on understanding the words of chromatin remodeling. So it's a pleasure to have Jerry here, and look forward to your talk.